Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Updates on Human Abuse Potential Studies, Challenges and Regulatory Guidelines. My name is Sonia Hunt, and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. At this point, I'd like to thank Dr. Vince Clinical Research and Altrios Research Partners who developed the content for this presentation. Dr. Vince Clinical Research, DVCR, is a full-service contract research organization specializing in early phase trials in both healthy normal volunteers and patient populations across a wide range of trial designs and multiple therapeutic areas. CRO services include project management, data management, biostatistics, monitoring, PK/PD analysis, medical writing, as well as site feasibility and management for multi-center trials. Additionally, DVCR operates one of the most innovative and technologically advanced clinical pharmacology units in the world. By leveraging technology and one of the country's most experienced leadership teams in early clinical development, DVCR provides smarter, faster data to their biopharmaceutical clients. And now, Altrios Research Partners provides strategic scientific consulting in the areas of abuse potential and abuse deterrence, CNS drug development, clinical pharmacology, and neuroimaging. Their scientific team includes Dr. Carrie Shodell, Dr. Megan Schwab, Dr. Naima Levy-Cooperman, who collectively have over 50 years of addiction research, pharmaceutical, and regulatory experience. Now it's my pleasure to introduce Dr. Brad Vince, the CEO and Medical Director of Dr. Vince Clinical Research. Dr. Vince has more than 20 years of experience in the clinical research industry. He has served as an investigator in over 700 clinical trials, including NIDA-funded human abuse potential studies, as well as over 40 human abuse liability studies. Now it's my pleasure to pass the mic over to Dr. Vince to introduce our speakers. So Dr. Vince, when you're ready, you may begin.
Hi, thank you, Sonia. Hi, Brad Vince. Thank you, everyone, for joining our webinar talk. I'm going to speak quickly to get through the introduction so we can get on to the content. First, wanted to introduce Dr. Naima Levy-Cooperman. I've had the pleasure of working with her for over 10 years as a colleague. Dr. Cooperman obtained her doctorate in neuroscience from the University of Toronto. She has over 15 years experience in CNS drug development, behavioral, as well as neurocognitive assessments, as well as the critical review and evaluation of abuse clinical trials. She's been involved in over 75 phase 1 clinical trials, and her team have extensive experience, again, in the critical review of abuse liability, abuse potential. Our next speaker will be Dr. Carrie Shodell. I've also had the pleasure of working with her for many years. She is a director at Altrios Research Partners. She completed her PhD in pharmacology at the University of Toronto, specializing in nicotine pharmacokinetics, as well as addiction. Dr. Shodell has extensive experience in clinical pharmacology, CNS drug development, including the assessment of abuse liability in both preclinical and clinical studies. Dr. Shodell has contributed to over 100 research and review articles, book chapters, as well as scientific presentations. She's also been involved in the preparations of numerous NDA as well as EMA drug submissions, and I could keep reading and reading. Very impressive CV. Thank you, Dr. Shodell, for joining us. And then lastly, I'd like to move on to my former competitor and current colleague, Dr. Lynn Webster, someone that I admire greatly for his contribution to this space. Dr. Webster is an internationally recognized clinical research expert in pain as well as addiction. He obtained his Doctor of Medicine from the University of Nebraska and completed his residency at the University of Utah in anesthesia. As our executive VP, Dr. Webster brings a significant amount of depth and invaluable expertise to our team. With more than 350 clinical trials under his belt, Dr. Webster has been an investigator as well as emerged as a leading figure in the field, contributing extensively in the medical review and literature with 300 publications, over 370 total publications and presentations. He's boarded in anesthesia, pain medicine, addiction, as well as a peer reviewer for NIDA grants, and as well has served as a human abuse potential research advisory committee member for more than a dozen times. So without further ado, we'll go ahead and begin our webinar presentation. Thank you for joining, and we'll turn it over to you, Dr. Naima Levy-Cooperman.
Thank you, Dr. Vince, and welcome, audience. Good afternoon. Today I'm going to be discussing new developments in the design and analysis of human abuse potential studies from the clinical perspective, with a specific focus on discussions held at the recent Cross-Company Abuse Liability Council, or CCALC meeting. I'm going to begin by providing a very brief overview of the CCALC meeting to provide some context to the latter discussions. I will also provide a review of elements of the human abuse potential study. First, when are these studies required? And then a summary of the design, endpoints, and analysis. The majority of this talk will focus on the topics of discussion at the CCALC meeting. The first topic was addressing discordant findings between clinical, non-clinical, and epidemiological data. The second topic was related to updates to the statistical analysis of these studies. The Cross-Company Abuse Liability Council was established in 2006 as a platform for interested parties, which included members of industry, regulators, academics, and public policy advocates, to come together and discuss the evaluation of abuse potential. Since 2006, there have been 7 dialogue sessions, the most recent of which occurred in Washington this past September. The meeting occurred over 2 days and included presentations as well as panel discussions on human abuse potential studies, abuse-related adverse events, behavioral economics, psychedelics, physical dependence and withdrawal, and non-clinical assessments. For this webinar, Dr. Shadell, Dr. Webster, and I will be focusing on a summary of the discussion on human abuse potential studies, adverse events, and physical dependence and withdrawal. 2 members of the Controlled Substances staff of the FDA presented on topics related to human abuse potential studies. Dr. Sylvia Calderon spoke to addressing discordant findings from non-clinical, clinical, and epidemiological data using examples from antiepileptics and dual orexin receptor antagonists, or DORAs, while Dr. Ling Chen presented updates and recommendations on the statistical analysis of human abuse potential studies. She provided recommendations on how to address non-responders or outliers, as well as suggestions to further enrich the subject population to improve study validity. But before we discuss these 2 topics, I'm going to provide a quick review on the design of human abuse potential studies. Drug abuse potential is determined from multiple sources of data. This includes the results of studies investigating chemistry, pharmacology, pharmacokinetics, animal and human behavior, abuse-related adverse events, as well as epidemiological information. The human abuse potential study is really just one component of the overall evaluation. So one of the most common questions that comes up is whether or not a human abuse potential study will actually be required. Generally, it's safe to assume that a HAP study is going to be a requirement for most CNS active new molecular entities, or NMEs. This is especially true for drugs that have chemical and pharmacological similarities to drugs that are already abused. Novel mechanisms of action that are not well understood will typically require an assessment, as well as products that show a signal in non-clinical studies, such as observations of sedation or locomotor activation in animals. The other signal that can trigger the requirement for a human abuse potential study is the observation of abuse adverse events in clinical trials, such as euphoric mood or hallucinations. These requirements were echoed and confirmed in Dr. Calderon's presentation at CCALC when she stated that, quote, "The need for a HAP evaluation is based on the identification of a positive signal in drug discrimination and self-administration, or the occurrence of abuse-related adverse events in clinical trials, or both," end quote. There are a few examples when a human abuse potential is not performed. Human abuse potential study is not performed, and this typically applies to drug products that have safety issues associated with the administration of supertherapeutic doses, which are required doses in human abuse potential studies. And it also applies to new molecular entities that clearly have no detectable CNS effects or effects of concern. Human abuse potential studies have 2 primary purposes. First, they aid in determining a novel drug's potential for abuse, that is, whether the drug is going to be scheduled or unscheduled based on the comparison of the investigational product to placebo. The second purpose is to aid in scheduling placement. This is typically referred to as relative abuse potential, and this is based on the comparison between the IP and the active control. In order to adequately assess a new drug's potential for abuse or risk of being used recreationally, these studies must include multiple subjective measures to assess various elements of the user experience, including positive effects, liking, negative effects, and effects associated with specific drug classes like stimulation or sedation. These measures are collected at multiple time points to ensure the effects are captured. There are multiple endpoints included for each measure, including peak and trough effects, area under the curve, and time to peak. The primary endpoint in all HAP studies is the peak score of the bipolar drug-liking VAS. This is used consistently across studies and is typically the score that is used to determine sample size calculations. In the past 7 years, inclusion of the 2 global scales of overall drug liking and take drug again as key secondary measures have been implemented. These scales evaluate the overall drug use experience because these scales are administered at the end of the day or the next morning, and subjects are sober when responding. Human abuse potential studies are typically randomized, double-blind crossover studies. They are designed as within-subject crossover designs due to the high variability of the subjective measures. Typically, multiple doses of the IP are included in order to investigate dose response, which is unknown in these products. So typically 3 doses of the investigational product, including a therapeutic dose and 2 supertherapeutic doses, while only 1 to 2 doses of the active control are included, as these products and their dose response or lack thereof are well understood. In most cases, we recommend these studies are conducted as inpatient studies, first to ensure that the subjects are not using other drugs of abuse while enrolled, and also to ensure subject safety, particularly after administration of these supertherapeutic doses. The studies enroll an enriched population of non-dependent recreational drug users who are history qualified, meaning that they must report having sufficient experience with the positive control drug class. The studies also include an enrichment design, which means enrolled subjects must successfully complete a pharmacological qualification that involves being able to distinguish between the positive control and placebo in a laboratory setting. By enrolling only responders, you limit the variability in the treatment phase and increase study power. Typically, these studies randomize between 30 and 60 subjects into the treatment phase with a pre-specified number of completers required. During the CCALC meeting, Dr. Calderon reviewed some instances in which drugs showed a limited signal in the non-clinical animal abuse potential studies. Both anti-epileptic drugs and DORAs have shown limited signals in non-clinical studies. In drug discrimination studies, neither class generalized or partially generalized the selected training drugs, and they also did not maintain self-administration in the self-administration models. However, for both the DORAs and the anti-epileptics, clinical study data had reports of abuse-related AEs such as euphoria and sedation, and the human abuse potential studies showed positive signals of abuse. So I'm going to provide a couple of examples that highlight first that animal abuse potential studies don't always predict results in humans, and also that highlight the range of human abuse potential results and how this may or may not impact scheduling. So on this slide, we see the results from 2 anti-epileptics, Lacosamide and Brivaracetam. In both studies, the comparator was alprazolam, a Schedule IV sedative. As a reminder, both the at the moment and overall drug liking VAS are 100-point visual analog scales where scores above 50 represent liking, scores below 50 represent disliking, and scores of 50 are neutral. On the top row are the drug liking and overall drug liking results for Lacosamide. The 2 doses of Lacosamide are presented in dark blue and light blue. As you can see, both doses of Lacosamide showed a positive response immediately after dosing that was higher than placebo and similar to alprazolam. However, this effect was generally fleeting and scores fell to neutral or disliking by 3 hours post-dose, particularly for the higher dose. These negative effects were then reflected in the overall drug liking scores. Remember, this scale is a global assessment that is asked at the end of day or next day when subjects are able to reflect on their entire experience. In contrast to the results with Lacosamide, all 3 doses of Brivaracetam had higher scores than placebo and similar results to alprazolam. In fact, the highest dose of Brivaracetam had higher scores than the high dose of alprazolam on both the at-the-moment drug liking and overall drug liking. But despite differing profiles, both these products ended up in Schedule V of the Controlled Substances Act. As a second example, here are the HAP results for Suvorexant, a DORA, which is used to treat insomnia. In this study, zolpidem, Schedule IV sedative hypnotic, was the active comparator. As you can see, Suvorexant in black had similar drug liking scores as zolpidem and showed no dose response. Scores on overall drug liking were greater than placebo and not different from zolpidem at all doses. Suvorexant was placed in Schedule IV. Both these examples presented in the last two slides emphasize 2 points. The first is that non-clinical data can be less predictive or inconsistent with HAP results for new molecular entities with novel mechanisms of action. This may be because the drug discrimination studies include only 1 or 2 training drugs, and new molecular entities may fail to generalize to these, but still show positive signal in a human abuse potential study. Also, there is unknown predictive validity of the self-administration model for drugs that do not belong to classes that are typically self-administered, like sedatives. The second point is that HAP study data are important for scheduling decisions, but variability within schedules is high. There are a number of other factors that contribute to scheduling decisions. For example, non-clinical data, clinical adverse events, safety issues, and of course, while pharmacological similarity is important, the product's indication and a regulatory precedent, as with antiepileptics, also matters. Another important point that was raised by Dr. Calderon was that post-market data, particularly preliminary or early epidemiological data on abuse, may not always be consistent with a positive HAP signal. Now, this may be a result of epidemiological lag, as well as other factors like availability and fads, utility of the product, as well as the fact that the drug itself is controlled, which may limit abuse. The second half of this talk, I'm going to be addressing some evolving elements of the HAP methodology as well as the statistical analysis. As I mentioned earlier, the qualification phase is required to identify responders or to enroll an enriched population. The qualification is typically a two-way crossover where subjects must differentiate between positive control and placebo. However, what we have seen is that often mean differences between control and placebo are approximately 10 to 15 points higher in the qualification phase compared with the treatment phase. This could be due to subjects reporting high drug liking for placebo or low drug liking for the active control. But if they passed during the qualification, what causes this discrepancy? It's likely that compared to the treatment phase, which is typically a 5 or a 6-way crossover, the 2-way crossover is a much easier test because there are only 2 arms. The subjects have a 50% chance of guessing correctly. This could also be related to insufficient subject training, or it could simply be that once the subjects pass the qualification and are enrolled in the main study, they are less motivated to respond appropriately. This discrepancy is problematic when considering that the predefined margin for study validity is 15 points, and there have been some validation failures in a few studies due to inappropriate placebo or active control response during the treatment phase. A possible solution that has been discussed is to change the design slightly and make the qualification test harder. A few options include adding a placebo arm for a 3-way crossover. This approach reduces unnecessary exposure to active drug product, but would need to ensure that subjects are blinded to how many doses of each product they will receive. A second option would be to include 2 doses of the positive control, a high and a low dose, or simply repeat the same dose. The other possible option is to ensure and to provide additional training at the site to ensure that the subjects understand the importance of both phases of the study. The second topic that Dr. Chen discussed during her session is really one of the solutions that has been implemented to address the last few issues around qualification criteria and not passing the study validity test that I mentioned. Since 2019, FDA has required that the study pre-specify a modified completer population. There have been a number of criteria proposed in the last 5 years, but at CCALC, Dr. Chen proposed that subjects would be excluded from the MCP population if during the treatment phase, their drug-liking VAS peak or Emax score for the active comparator was less than or equal to 55, so essentially not responding appropriately to the control drug. Their placebo drug-liking Emax was greater than their active control Emax, so either a placebo or an active control non-responder, or if their peak scores across all treatments did not differ by more than 5 points, so showing similar scores across treatments. One of the benefits of this approach is that you are only enrolling subjects who responded accurately, thereby increasing the likelihood of an accurate assessment of the study drug's abuse potential. There is also less risk that the study will fail based on the validity test. In Dr. Chen's talk, she presented data in which she retroactively applied the modified completer population criteria to 3 studies that had failed based on the validity test. and she found that between 13 to 28% of subjects would have been removed from the analysis, with stronger results observed. In addition, she stated that removing these subjects would not result in any bias in the treatment comparisons. But there are also risks. Decreasing the sample size may result in less power if a large number are excluded. It may also be difficult to determine how many subjects to enroll to ensure enough completers. And of course, as you eliminate subjects from the analysis, you are likely to be left with an incomplete Williams square, which may cause issues with your first order carryover effect analysis. In general, HAP studies are useful in informing scheduling decisions, but the totality of the data is what will ultimately determine a drug's schedule. And while HAP study designs overall have not changed dramatically, the statistical analysis and methodology are constantly evolving to account for some of the issues that come up as more of these studies are conducted. As I mentioned earlier, it's been recommended that in order to improve the enrichment design, we could shift toward a more complex pharmacological qualification. But the question is, will this added complexity be worth the additional subject and logistical burden? So additional analyses of whether or not there are shifts or improved performance with the more complex qualifications will need to be conducted. We also recommended that the study sites selected train their staff and subjects, emphasizing the difference between qualification and the treatment phases, as well as ensuring that subjects are adequately trained in all subjective measures. Finally, because the modified completer population is now a requirement per FDA, we suggest, if possible, calculating sample size data based on more recent studies in which the MCP is used in all analyses. Thank you very much for your attention. I'm now going to pass it on to Dr. Carrie Fidel, who will be discussing abuse and dependence-related data from clinical safety and efficacy studies.
So as Nama mentioned, today I'm going to be talking about collecting abuse and dependence-related data from clinical studies. And what I mean by safety and efficacy studies, of course, is the whole gamut of phase 1 to phase 3 clinical studies. I'll be addressing two topics today. The first is the potentially abuse-related AEs, and the second is the drug discontinuation-emergent AEs, or DDEAEs. So when we're talking about abuse-related AEs, we can consider them to be AEs of special interest, or AESIs. We need to systematically categorize, tabulate, and analyze these AEs. These AEs are one of the only opportunities that we have in development to collect evidence of abuse potential in the intended patient populations, as well as healthy volunteers. These AEs are used both to determine signals in clinical development, which may require further abuse assessments, but they will also be assessed as a part of the NDA submission in the determination of scheduling of the drug. The data for these AEs are derived from all clinical studies, and typically this would be split into healthy volunteers, patients, of course, the primary indication, but also if there are any other indications that are pursued in clinical development, even if they're not being submitted in that particular NDA, the data will also need to be reported for those studies. And then finally, we'll present the data from recreational drug users from the HAP study separately if a study was performed. Now, these types of AEs primarily assess subjective effects of interest. And then specifically, this would be mood elevation, sedation, stimulation, or hallucinogenic effects. And this is directly from the US Controlled Substances Act, which indicates that if you see such effects in clinical development, you need to submit an abuse potential evaluation. Now, in our experience, the events of particular interest are those such as euphoric mood, feeling drunk, feeling abnormal, or really any type of hallucination. We also look at things like sedation and stimulation, but this is usually only of interest if these events co-occur with the events of particular interest. For example, the sedation and euphoric mood co-occurring in a single patient. The other type of data that we can collect from AEs are the so-called aberrant behaviors, which might signal actual cases of abuse or dependence. The issue with these, of course, is that these events are very rarely reported in clinical trials. So at the CCALC 2023 meeting, Stephen Galati presented an updated list of abuse-related AE search terms. It's not dissimilar from lists that we've seen in the past or the AEs that are defined in the FDA abuse potential guidance. However, it is more concise and more clearly defined. And as we are happy to see that there was no AE of dizziness on this list, as this AE occurs very commonly with CNS drugs. Now, investigator site and monitor training is critical in the reporting and collection of these AEs because they do represent mainly subjective types of experiences. And because of that, there's many opportunities for misreporting. And some of the issues we've seen with the subjective AE reporting, one is that we have seen sometimes investigators not reporting something like euphoria or euphoric mood because they don't consider it to be adverse. We've also seen cases where a subject will actually report multiple different AEs that tell us something about the pharmacology of the drug, but those different experiences get collapsed into sort of a single diagnosis, and we lose that, the pharmacological knowledge that we can gain. We've also seen some interesting interpretations of a subject's description by investigators or site staff. And then finally, also at the level of coding, when we code the clinical terms to the MEDRA preferred terms, we've seen some interesting adventures in miscoding. My favorite being a subject who reported that they were feeling hungover, and somehow this got coded to an AE of feeling drunk, which, as I mentioned before, is an AE of particular interest. Now, the other type of event that signals more of an aberrant behavior or actual abuse or dependence, as I mentioned, they are very rare, and you're not going to get a subject or a patient that's directly reporting these events. They're not going to say they feel addicted or they're abusing a drug. So these events actually do require a kind of diagnosis by the investigator based on their judgment of the patient's behavior and their history. And as I keep reminding everyone, this type of AE should be treated as an SAE, and this comes directly out of the Code of Federal Regulations, where these events are noted as AEs of medical interest and should be provided with full case narratives. Now, some of these AEs are going to require narratives to be submitted in the NDA submission. In Stephen's presentation, he provided an updated list of AEs that will require narratives in the submission. In general, we find that the list is quite reasonable. It has things that you would expect. There were a few questionable inclusions, for example, somnolence. And when I questioned the inclusion of somnolence specifically, because we see it so commonly reported with any CNS drug, it was clear two things. One, the AE, as I mentioned before, the CSS is interested in clusters of events, so whether somnolence is co-occurring with other events of interest. And the other issue, of course, is CSS is well aware of the reporting and coding issues that can occur in clinical trials. And so I think that part of the requirement for narratives is to try and get around these issues and really to get at what's actually happening with the patient. And for that reason, I would again emphasize proper investigator and site training. And another thing that we can do is actually database the subject's verbatim description of the events wherever possible, because that can really give us a good idea of what's happening, what's actually happening. So Stephen also presented an analysis of abuse-related AEs that was conducted by CSS, comparing 10 scheduled and unscheduled drugs from selected studies. And as they found that there were more abuse-related AEs observed with scheduled drugs compared to unscheduled drugs, and that these differences were most prominent in the healthy volunteer studies. And this isn't surprising because in these studies, we don't have the confound of underlying symptoms. There's usually few or no conmeds. And then, of course, some of these studies will use supratherapeutic doses, which allows an evaluation of dose response. And for that reason, training and a selection of appropriate phase 1 sites is particularly important, because you don't want to end up with a signal early in development that's going to cost you later on. So this is actually our own retrospective analysis of abuse-related AEs, and these are from HAP Studies. As you can see in the first panel, AEs associated with mood elevation are quite high with opioids, and this is primarily euphoric mood, as well as some of the other classes. Sedative AEs are high with sedatives, and stimulant AEs are seen with stimulants. And then we also see a little bit of hallucination or perceptual disturbances type of AEs. And this analysis does show that proper collection and analysis of these AEs is actually useful for determining pharmacological effects of interest. Here's another analysis that we conducted looking at the incidence of euphoric mood and the correlation with peak drug liking from HAP studies. As you can see from this initial panel, there's a very strong correlation between subjects reporting euphoric mood and reporting drug liking for the opioid class. However, if we extend the analysis to include other drug classes, including a number of unscheduled drugs, which were included in some HAP studies as negative controls, the correlation, while still statistically significant, is quite a bit lower. So we have to keep that in mind, that some of these AEs are particularly useful for certain classes and potentially less useful for others. So now I'm going to talk about the DDEAEs. Now, these drug discontinuation-emergent events are those AEs that emerge following discontinuation of a drug. Obviously, this refers to multiple dose or chronic dosing studies, typically using a cutoff of at least 7 days. These AEs provide data on physical dependence potential in humans, and this is considered both in the drug scheduling determinations according to the Controlled Substances Act, but it's also a clinical safety issue for patients when they're discontinuing a drug. So unlike the abuse-related AEs, for this analysis, it's really the collection of any AE that occurs post-discontinuation of the drug. For some indications, abrupt discontinuation is contraindicated. In that case, we would simply summarize the taper emergent events and potentially any AEs that occurred in patients who had abruptly discontinued. And particularly in these situations, we may need to rely primarily on other data for physical dependence, such as that collected from animals or healthy volunteer studies. Now, these AEs can be quite difficult to interpret because particularly if you have a longer half-life drug, some of the events may actually reflect residual drug effects or the return of the underlying disease symptoms. For that reason, we compare not only the active drug to placebo, but also we compare the off-treatment or the DDEAEs to on-treatment AEs. And this was confirmed in Emily Deng's presentation at CCALC. To define on and off treatment AEs, we'd use the half-life to determine a cutoff. Usually this is at least one half-life, and practically it's often the next day or even the following day, simply because in the later stage studies, the time of the events are typically not recorded. Now, these events tend to occur somewhat sporadically in the off treatment, so you'll often see that these AEs occur with a lower incidence compared to on-treatment AEs. And this may simply be due to the shorter duration of the follow-up period compared to the treatment or maintenance period, so you have less opportunities for reporting the events. One way to adjust for this would be potentially to look at an exposure-adjusted analysis to sort of equalize the treatment durations. However, we found that because of the very short durations of the follow-up periods, for example, they may be only 2 weeks or a month compared to a 6 or 12-month treatment period, that sometimes even AEs that occur in a single patient can appear to be inflated in this analysis. And so for that reason, what we typically do instead is we'll look at the whole picture of the on-treatment to off-treatment AEs. And what we expect to see is a decrease from on to off treatment, and anything that actually goes up can be treated as suspicious. And the other important thing to evaluate is not only looking at a single AE, such as headache, that occurs in a few subjects, but whether these AEs are actually occurring as clusters within an individual patient. So obviously, the latter situation may indicate the presence of an actual withdrawal syndrome, and that's going to inform how clinically relevant the physical dependence potential is, both for patients, but also from a drug-seeking or drug-taking behavior perspective. So here's an example of some off-treatment DDEAEs, and this is actually real data that's been modified for this presentation. As you can see, there's a whole bunch of AEs that occur with a higher incidence with the active drug compared to placebo in the off-treatment period. However, if we look a little closer, we see that some of these are probably not related to any withdrawal syndrome. They're things such as viral infections or laboratory abnormalities. But even if we remove those, there's still a fair number of AEs that occur with a higher incidence. Now, if we only looked at it this way, we would probably conclude that this drug was associated with a withdrawal syndrome. However, if we compare to the on-treatment AEs, we can see that in this scenario here, all of the AEs are either decreasing sharply or staying stable from on to off treatment. In a different scenario, we can see there's actually a little cluster of events that increase a little bit in the off-treatment period. And if we look at those more carefully, we can see that they're primarily stimulant type effects, which is of great interest because this is a sedative drug. And when we're looking at a withdrawal syndrome, we often see that it may be presenting with opposite pharmacology to its on-treatment. So in conclusion, as mentioned by Stephen at the CCALC talk, these AEs do not provide a definitive signal of abuse potential, but rather form a component of the full abuse potential assessment. Abuse-related AEs support the determination of subjective effects that may be of interest for abuse, and they provide early signals which can be used to determine the need for further evaluations, such as in a HAP study, and as well provide the opportunity to evaluate abuse potential in patient populations. The post-treatment DDEAEs provide support for animal studies of physical dependence, and as well as prospective clinical assessments. And with that, I'll pass it over to Dr. Webster, who will be discussing these issues. Thank you.
Thank you, Carrie. It's a great sequence into what I'm going to be talking about, which is the withdrawal symptoms following or a part of the human abuse potential studies. As I said, my talk is going to discuss the evaluation of withdrawal as it pertains to human abuse potential studies and its application perhaps to the labeling. And differentiate between acute and chronic withdrawal symptoms and their severity, and maybe how and when we should be monitoring to detect the short-term versus the long-term effects of drug discontinuation. This slide represents the different components that need to go into an evaluation of a HAP and perhaps the scheduling, as both of the previous speakers have indicated. But one of the key elements is trying to understand the discontinuation or the withdrawal behaviors once the medication has been discontinued. Withdrawal and physical dependence evaluates whether the chronic administration of a drug at a therapeutic plasma level produces withdrawal symptoms upon discontinuation. There are some key factors that contribute to this. These are not the only ones, but the binding of the drug molecule to the receptor, that is the strength of the binding, the timing and duration of the dissociation from the receptor, the concentration and the duration of the drug compound on the receptor. And some of these are limited in clinical trials, so they may not necessarily apply to the real world after a drug has been exposed to a patient for an extended period of time. But these are key in trying to understand the mechanism of action that can contribute to withdrawal. Now, there are traditionally short-term, and Carrie just mentioned basically the evaluation of the discontinuation behaviors and some of the signs and symptoms, but there are short-term and long-term signs and symptoms. Generally, the short-term are physiologic symptoms, and these are usually days or hours to days, whereas the psychological symptoms occur usually over a longer period of time, weeks to months. And they're very specific with the drug class. We have to use certain scales depending upon that drug class, and Carrie had indicated that sometimes we can't perform withdrawal studies because of the pharmacology of the drug that places some patients at risk. Traditionally, it is the visual analog scale that assesses these symptoms, certainly in the acute phase or in the short-term phase. Some of the dependence evaluation depends upon a diary, and during clinical trials, it's mostly collected through an e-diary of some type, and yet this is also the physiologic and cognitive behavioral assessments can be a little different. They both can occur early, but we see a difference in the severity and the magnitude of these over a prolonged period of time. Sometimes we want to get PK data. Generally, that is for the short-term evaluation and not for the long-term, although it could be, and associate that with the withdrawal signs and symptoms. But we also know that many withdrawal signs and symptoms that are extended can occur much longer after the drug has been eliminated from the body. There are a few drugs, for example, that have developed a physical dependency like beta blockers or monoamine reuptake inhibitors that don't have any significant withdrawal symptoms. The label does want to understand the effect of the discontinuation or the withdrawal effect. And there are three major areas, as has been previously mentioned, that is whether or not it should be scheduled, whether or not there is abuse, but the dependence or the physical dependence of it can precipitate, or the knowledge of it is important, because if there is a significant amount of adverse effects with the discontinuation or with the withdrawal, that can lead to behaviors that can be very harmful for patients. For example, somebody who's dependent on opioids and then they go into a great deal of pain, nausea and vomiting, diarrhea, that can lead to a reemergence basically of their substance use disorder. So the abrupt withdrawal is likely to cause significant AEs in healthy subjects, physical dependence studies, as well as in animal models that must be considered in the label. Now, there are a lot of common variables among withdrawal studies, and I have those listed there, but these may all change in their severity and their magnitude depending upon how that withdrawal occurs. Is it a precipitated withdrawal with an antagonist of some type, or is it a tapered withdrawal? Is it a rapid tapered? And the importance is that we need to understand how this will affect a patient once a drug is approved, and that would have to be in the labels, because the label then could give instructions to the clinicians as to how to prevent this from happening or how to treat it. I inserted this slide because I think it's interesting to show a timeline, at least for the opioid withdrawal scales, because all of the withdrawal assessments are based upon some kind of a scale that is drug specific. But with the opioid, it started back in 1938 with a Hemmelsbach scale. And over a period of time, these are for the most part, most of these scales have been modified somewhat from the original one. We use, though, the Hemmelsbach scale a large amount for the evaluation of withdrawal from peripheral opioid antagonists when they were being developed. But today, primarily, opioid withdrawal signs and symptoms, and the COWS is the primary scale that is used, at least for opioids, but there are other scales, as I have here as an example. With cannabis withdrawal, you've got the marijuana withdrawal scale or the cannabis withdrawal scale. Each of these have to be used at various times in order to detect the magnitude and severity of the symptoms. And at minimum, they need to be at the onset, the peak of their effect, and the duration. Peak usually means the most dramatic change or delta of the receptor exposure to that particular molecule. I have an illustration here of cannabis, for example. You can see the magnitude of the various symptoms occur at different times, the severity, and that the severity really changes from physiologic to psychological terms. And this is with cannabis over about a 3-week period. But of course, we know that with cannabis withdrawal, that it can be protracted, it can be for months and maybe even years. In a different way to look at this, and sometimes we're challenged with this type of an evaluation, but it's important for the documentation that an observer may feel and record through a scale one finding that is different from what the patient or the subject, I should say, is actually experiencing. So you see on the left, the observer sees that a dramatic decrease in the withdrawal severity very quickly. But when the subject was asked, the severity did not decrease over several days. And this may be true particularly with the cognitive changes. There was a recent study to illustrate this that was published that had more than 1,200 benzodiazepine users, and they included people who were taking benzodiazepines, who had been tapered off of benzodiazepines, and then those who were just gradually tapered, fully discontinued and gradually tapered. And what was the important point to this, in my view, was that the symptoms and the duration lasts over a year. So adverse events may not be collected in our current structure or designs of trying to detect the signs and symptoms of withdrawal from some of the medications that are being studied. The withdrawal symptom identification, it's unique, as I say, and Carrie had already mentioned that sometimes we're looking for the opposite effect of the desired effect. That is, for example, opioids produce analgesia in their therapeutic indication, but when there's abstinence or there's a withdrawal from an opioid, it may induce pain. We may have to understand the mechanism of action then and apply that to the design of any withdrawal study that is either short-term or long-term in order to get really a good profile of how that drug is going to behave once it would be in the community. So the severity scales could be skewed somewhat because it could be disease related, and Carrie had just mentioned that, or it could be drug related. And so we have to take into account also the patient reported outcomes as well as the normal pharmacokinetic and vital signs. Factors that can impact the withdrawal severity obviously is the duration of the drug, the concentration of the drug, the underlying comorbidities, particularly if there are some mental health and physical health conditions, and whether or not they've been in treatment adherence, as well as whether there is a polysubstance abuse problem that can confound the interpretation of the data. This is in the real world, and it's often not something we see in our clinical trials of evaluating drugs in relatively healthy patients. This slide shows that the variability in the acute or the short-term evaluations based upon the mechanism of action. It still could be up to 2 months, anywhere from a few days to a few months. Some of the other factors impacting withdrawal duration, as I say, mental health, but the route of administration, the duration, frequency of the drug that's being used, it doesn't have to be substance abuse, just any drug. The severity of dependence, which is really the upregulation and that can be dependent upon the concentration of the drug and the time at which it's exposed to the receptor. So the chronic withdrawal symptoms and duration are something that the FDA has not traditionally looked at beyond the first few months, except in some of the phase 3 trials, but then it's still limited probably to a year. Depression is one of the most common withdrawal symptoms with many of the drugs, particularly with the GABA agonist or GABA receptor involvements. There is some interest in trying to assess the long-term effects of drug withdrawals with now telehealth and mobile health if the e-diaries are not effective. And the advantages to this is that the data can be immediately available, and if there are problematic behaviors, there can be quick interventions, because the current technology that has just evolved since COVID, frankly, has allowed us to have a more direct contact with our subjects or with our patients, if it happens to be something that's been approved, the drug approved, so that we can modify our treatment and intervene before it becomes problematic. And so that means that we can assess several times during a day. We can assess chronic symptoms over a week, or maybe we can take assessments on a monthly basis, and it can last, particularly for the psychological assessments of withdrawal, can last 6 months or a year. I'll turn it back to Sonia now, and thank you for your interest.
Well, thank you very much for that presentation once again. Now we're going to begin the Q&A portion of this webinar. And now I'm just going to open the room up for the Q&A portion of the webinar. So now I know that we did receive a bunch of questions while you were speaking. So let me start with this first question here. We'll try to squeeze 2 questions in with the time that we have together. So the question I have here from this audience member is, you suggest that the timing of the withdrawal assessments is important and frequently not obtained at the correct times. When should the assessments be obtained? Who would like to take this question?
Sonia, this is Lynn Webster. I'd be happy to take it. Yes. Often we have a designated time, or we choose maybe a daily assessment to collect the information, and that might be inadequate because we're not matching that then with the time at which you might see the greatest withdrawal symptom or signs. But I think and that's with the acute withdrawal. More importantly, I think we're not collecting data on patients that or subjects that have been exposed for a long time, 6 months or a year in phase 3 studies, and measuring it for a month or 3 months following the discontinuation, because we know that many of the drugs that we study have long-term effects, either from an acute withdrawal or from just a discontinuation. So I think we need to rethink about how to identify the withdrawal signs and symptoms in a way in which it can help clinicians treat their patients and prevent this from happening or causing harm to them.
Oh, thank you very much for that very detailed answer, Dr. Webster. Now I'm now going to go to our next and final question. This audience member is asking, why should withdrawal assessments extend beyond the acute withdrawal phase? How will this information help clinicians? I guess this is a question again for you, Dr. Webster.
Yeah, well, I think Carrie addressed this too. I mean, this is something that the FDA is interested in, but if we don't collect that information, then we won't be able to know how to advise clinicians in a label how to, 1, be aware of it, or when they should be aware of it, and then 2, how to treat it and prevent it from occurring.
Okay, thank you very much for that, Dr. Webster. Well, thank you very much for those answers. We have reached the end of the Q&A portion of this webinar. For those of you who are interested in hearing more, stay tuned because more webinars will be coming up. And I'm going to send you a link in the chat box right now, so you can also check this link out to be up to date on when the next webinar will be, and also check out xtalks.com. Okay, now if we could not attend to all your questions, remember the team at Dr. Vin's Clinical Research will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see there on your screen, info@drvince.com. Now, please join us in thanking our speakers for their time here today. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.